Okay, today we will talk about uh, a part of mammalian physiology. This is going to be a DSE paper, discipline specific elective. Most probably, what I hear that mm, the teachers are concentrated for mammalian physiology and wildlife conservation and management, not aquatic biology. But even if somebody takes aquatic biology, just let me know. We will cover that also. If some colleges if Asansol girls take, so far as I've talked with Supriyo, and they are also thinking about mammalian physiology and wildlife conservation. But if they take any other thing, just let me know. Then we will have that also. So.
Okay, sir.
Hmm. So far as I my information knows, when I talked with Supriyo, he told me that it's basically we were of the same opinion that these two subjects will be better, because human reproductive biology, it's absolutely mm, uh, uh, something. It is first time it will be introduced, and many teachers and students, they may not have the notes and make appropriate materials, although they have studied parts of it. And as for aquatic biology, there are no really good books, and getting materials are difficult, but it was there in the former times. So that can be a chance that there will be aquatic biology and mammalian physiology instead of wildlife conservation and management, because both of them are old. So this is actually how students will elect, and that is going to be taken up. So for that, we can have another class, and that should not be a problem. Whatever be it, the discipline specific electives, only one paper which really is very sort of understanding, or you need to do properly in the discipline specific, and that is this mammalian physiology. There is a bit of understanding which you need. As regards CC, for your core courses, it will be common everywhere. You have biostatistics and bioinformatics. This is actually in the CBCS. This is the first time it is being introduced in this LSCF. So biostatistics is something which you have to practice. You cannot rely exclusively on bioinformatics, because some marks of biostatistics you need to answer. So the questions will be coming from there. And economic zoology is easy, that paper. But still, nonetheless, it is a scoring paper. So you must try to get as much marks as possible. Okay. So today, from mammalian physiology, the syllabus has not yet come. Do any of you have any syllabus of mammalian physiology? I don't have it. So,
Yes, sir.
Yes,
uh,
sir.
then send it to me, please, later. Okay, but I know this nerve impulse is going to be there, muscle is going to be there, digestive system is going to be there, things about circulatory system, everything is going to be there. This is for sure. And so to start with, I will start with nerve impulse, and that is something in all universities. It's going to be uh, actually it's going to be a very important question. Nerve muscle is a very are very important questions. Apart from that, kidney, glomerular filtration rate and all these things, kidney filtration is important, and some components of blood that can be good. Those things you may are, and about heart functioning. So behind every behind everything, you have nerve impulse is the first thing which it's best to study. You can have skins, tissues, those are easy chapters, but the difficult chapters are the nerve impulse, muscle contraction, and I would say kidney filtration. These are the three most important, I would say. You must try to finish them first: nerve, muscle, your kidney, the excretory system, in my opinion. And then we will see what is important. As the semester progresses, we will understand what is important. So I'm going to share my screen. Okay, share. So nerve impulse, I've already have my presentations prepared. So are you seeing a white screen?
Yes, sir.
Oh, great, great. सफेद सादा. किसी को कुछ मालूम है क्या? नर्व इम्पल्स है या दिमाग ऐसे सफेद है? डू एनी ऑफ यू नो अबाउट नर्व इम्पल्स, और इज योर ब्रेन white on this? শুভজিৎ কিছু জানে না, ও তো ফেল করবে। এন্ড বাকিরা? শুভজিৎ এই সেমিস্টারে ফেল নাকি পাস?
Sir, same thing, different semester.
সেম থিং ডিফারেন্ট সেমিস্টার, আচ্ছা। ওকে, সো লেট আস টক অ্যাবাউট নার্ভ ইম্পালস। The first thing which we are going to study is the different cranial nerves. So, what are cranial nerves? Can anybody of you tell what are cranial nerves? Just briefly, aram se, but you have to say the right answer. That is not. What are cranial nerves? You have done some dissection, I think. What are cranial nerves? কেউ জানো? শুভজিৎ? হ্যাঁ, পর্ণালী বলো, বলো পর্ণালী, বলো কে বলছিলে বলো।
Sir, the nerves which are arises from brain.
Exactly, the nerves which are coming, main nerves which are coming from, originating from your brain or from the cranium, inside the cranium. And these nerves, they go throughout the body. So 12 important nerves, all vertebrates have these important nerves. They cover throughout the body. As for nerve, for most of the important activities of the body, the 10th nerve is very important, the vagus nerve. So you have to know the name of all those nerves. So this is the brain, তলার থেকে, from bottom. You remove the brain from bottom, and these are the little nerves which are coming out from here. এই যে nerves গুলো হচ্ছে, I have not given the names. So you can see this nerve over here, this nerve, this nerve, this is a green color. They are all given in different colors so that you can understand that they are originating from different places. So the first one is olfactory nerve. This is the first olfactory, this is the second nerve. So in both half of your brains, you have an olfactory nerve. It comes out like this. It goes to your nose and you can smell and sense. This is a very famous nerve. Let us see what it is. The second nerve is optic nerve. তোমার চোখে, it goes to your eyes. It just below the olfactory, that originates. Next is oculomotor. Can anybody of tell what is oculomotor? What does the oculomotor do? Okay, anyway, we will talk about it. Then you have trochlear. It goes to your ears. Trigeminal. You're biting and all these things. These trigeminals, they help this nerve. They give the impulse for the muscles to move. Then you have abducens. Facial, from your face. Goes to your face. Vestibulocochlear. Glossopharyngeal, very important nerve. Hypoglossal, also a very important nerve. Vagus, the most important nerve. Accessory. All these cranial nerves, these 12 pairs of cranial nerves, you have to know the name. Olfactory, oculomotor, optic, trigeminal, trochlear, abducens, vestibulotrochlear, facial, glossopharyngeal, hypoglossal, vagus, and accessory. So to remember all the names of these nerves, there is a very vulgar formula which I used to remember the names of these nerves. This I cannot actually, being a teacher, I cannot tell. So as a student, I can only you people can know. So what you have to do is, there is a book of Arthur Haley known as Doctors. So in this Doctors, in the first chapter, you will find that particular way by which you can remember these nerves. In fact, it's so vulgar that I can't even write it down. Okay. So let us look at the structure of a nerve. So the nerve first has got a nerve body. This is known as soma, right? And then it has got certain things called, this is known as axons, which are coming out. A fiber, a process which goes throughout. সাদা হয়ে নাবে, that is, yeah, can you see this axon, this process? This is known as axon. দেখতে পাচ্ছ?
Yes, sir.
Yes, sir.
Okay, okay. And you have the cell body. From the cell body, you have little processes coming out. These processes are coming out like branches of a tree. These processes are coming out. They are known as, can anybody tell what are they? They are known as dendrons or dendrites. Many of them dendrites. And you have a nucleus. Then you have something like a pyramid-like structure coming on. This is known as axon hillock. And from that, the axon starts to go, and it ends into what are these? In an axon terminal. The nerve ends in a, the axon ends in an axon ending, and it ends in a bulb-like structure, and that is known as synaptic bulb. So this bulb, it connects to the dendron of the next nerve. This dendron, say this part is open over here. This axon, this one bulb goes and fits over here. From this nerve, some neurotransmitters like acetylcholine is released, and then it stimulates the next nerve. From there, then the electric current passes, goes to the end, and then another nerve some starts, and this region is known as synapse. So this is actually, and many nerves, they have got a covering known as myelin sheath. So they are known as myelinated nerve fibers. Even then, there are fibers which are non-myelinated. So in between this myelin sheaths, you have little empty, naked exposed area of the nerves known as node of Ranvier. So electric current comes, jumps, and this is known as saltatory conduction. When the electric current, normally unmyelinated, the current passes straight. Here what happens? It jumps. The current is here, it jumps and comes over here, then it jumps and comes over here, jumps. So by jumping, this type of electrical conduction is known as saltatory conduction. So this was the structure of how a typical neuron looks like. So let us look at the anterior part of the neuron. So এটাকেই বড় করলাম. This looks big. So you can see these processes. These processes are the dendrites or dendrons. So these are the dendrites. These tree-like branching processes, they are known as dendrites. Then you have the body, gata. It's known as cell body or cell soma, S-O-M-A, soma. Some people call it carrion. Then the initial segment starts for the axons. Then this hill-like region which originates out, that is known as axon hillock. You have lots of mitochondria inside. They are mitochondria for energy. Inside of the cell, like all cells, it has got a cell cytoplasm. There are little bodies, granule-like structures known as Nissl bodies inside the nerves. You have the nucleus, and from where the dendrite starts, that is known as neurofibril. Now let us go to the middle part of the neuron. The axon starts. Inside the myelin sheath, there is a nucleus. They are known as nucleus of Schwann cells. So there are cells which cover, they are known as Schwann cells. So under the myelin sheath, you have the Schwann cells and the cytoplasm of the Schwann cells covering neurolemma. These little spaces, nodes of Ranvier. You have a neurofibril coming out from here. Axoplasm, axolemma, inside. Then the terminal part of the neuron. This is very important. So it ends in an axon terminal, and these little bulb-like structures are known as synaptic end bulb. Clear about the structure of the neuron? Yes, no?
Yes, sir.
Yes, sir.
Yes, sir.
Okay, okay. There are other support cells also. So let us see. This is one nerve fiber, this is another nerve fiber. Two nerve fibers are there. So along with that, the first is covering all the nerves are the Schwann cells, a gold blue color Schwann cells. They are also known as neurolemma cells. They produce the myelin sheath around the neuronal axons. Hence, it gives support to neurons in the peripheral nervous system. These Schwann cells, they produce the myelin sheath. Then you have cells known as oligodendrocytes. So they are a type of large glial cells found in the central nervous cells and produce the myelin sheath insulating the neuronal axons. So they produce these myelin sheaths along with Schwann cells. Then you have the microglial cells. They are a specialized population of macrophages that are found in the central nervous system. They remove damaged neurons and infections and are important for maintaining the health of the central nervous system. Then you have the ependymal cells. They are a type of neuronal support cells or neuroglia that forms the epithelial lining of the ventricles of the brain and the central canal of the spines. And you have the astrocytes. They are named because they are star shaped. They are the most abundant glial cells in the brain that are closely associated with neuronal synapses. They regulate the transmission of electrical impulses within the brain cells. So these are the other support cells. Then to classify the neurons, they can be classified as multipolar neurons. So the structure is clear. Bipolar, both sides. You have axon terminals coming out from both sides. And unipolar, it is present like this. This is the classification of neurons. A multipolar neuron is a type of neuron that possesses a single axon and many dendrites. Single axon and many dendrites. Allowing, this is the multipolar neuron. A bipolar neuron, you can see it. It has got two extensions, one from here, one from here. And a unipolar neuron in which only one process called the neurite extends from the cell body. This is the neurite. It is extending from the cell body. So very important before we go to nerve impulse, they are the ions and ion channels involved in nerve impulse propagation. So very important in nerve impulse during a nerve impulse, there are electrical conduction takes place, as well as chemical conduction takes place. So two types of nerve conduction. One is chemical with the help of neurotransmitter, and next is electrical current is passed. And how do these electrical current pass? They pass because of these ions. Ions are charged, positive and negative, they are charged because of these channels opening and ions being going out of the cell and or coming inside the cell. So the cell polarity, the membrane potential, or the cell electrical charge on the cell is changed. We will talk about it in shortly. So first thing, you have the potassium leak channels. This is on the cell membrane of on the axons. So potassium leak channels are present. So these channels allow passage of potassium. This channel, leak channel, why is it called? Because this channel randomly opens and closes. So you can see when it's closed, no potassium can come in or go out. When it is open, potassium, since the concentration of potassium inside the cell is very high, it starts to leak out. Similarly, you have sodium ion leak channels present. That is, they randomly open and they randomly close. Sodium is high outside, less inside. So when these leak channels open, then sodium enters. Then you have ligand gated channels. This ligand gated channel, this is closed. This is ligand means when something, this red color comes and binds, this open. This is right now closed. A chemical stimulus, like this chemical, red color chemical, most probably acetylcholine, it comes and it opens the channel. Channel like acetylcholine comes and binds, and then this channel opens and ions can come in and go out. These are ligand gated channels. Then mechanically gated channels. Closed. This is in a closed position. This channel on the membrane. A mechanical stimulation, a touch or something comes. It opens the channel. And this channel opens and then ions can come inside or go outside, whatever. So this is known as mechanically gated channel. You have the famous voltage gated channels. A change in volt current. Normally during resting, it is the, if you may put an electrode inside the cell and one electrode outside and attach it to a potentiometer measures voltage. So you will see the reading of minus 70 millivolts coming. So it is not 0, 0. So charge, there is a difference in charge. So membrane, change in membrane potential opens the channel. So when the voltage falls to minus 50 millivolts, suddenly this channel will open. For some reason, because of leak channels or whatever, or some touch or mechanical gated channel, the voltage changes, these channels will open. These are voltage gated channels. Clear so far about the different channels?
Yes, sir.
Sabai, everybody?
Yes, sir.
Okay.
Yes, sir.
Yes, sir.
Okay, okay. So membrane potential of neuron at rest is minus 70 millivolts. Why is it like this? So this is a axon. This is the axon. Normally, if you take a voltmeter or potentiometer, you put one electrode outside, one difference inside. So potentiometer, it measures the potential difference or voltage. So charges flow from high potential to low potential. So if you measure it, the difference is, you will get a difference of minus 70 millivolt in this part of the neuron, one microelectrode inside and one microelectrode outside. the ground electrode outside, you will get a minus 70 volt. So this is the resting membrane potential of a nerve. So what is a nerve impulse? A nerve impulse in neurons, it is also known as action potential. Action potential ja nerve impulse tai. What happens in nerve? It is a sudden reversal of the electrical charge across the membrane of a resting neuron. The reversal of charge is called action potential. So outside is plus, inside is minus, therefore it comes minus. Suddenly inside becomes plus and outside becomes minus. There is a change in membrane potential and then an electric current flows from one potential to another. So it is a signal which is electrical and it is transmitted along a nerve fiber. So what are the components? It consists of a wave of electrical depolarization that reverses the potential difference. We have seen outside positive and inside negative. It will change. Inside will become positive and outside will become negative. That reverses the potential difference across the nerve cell membranes. It is also associated with corresponding repolarization and hyperpolarization. We will talk about them. There is a difference between action potential, minor difference, and nerve impulse. Action potential is the electrical polarization of the membrane of the nerve. So in action potential, there is a difference. And nerve impulse, this action potential starts to move along the nerve fiber. But the action potential and nerve impulse, they are used synonymously. একই বলে সবাই দুজন ওটাকে. So this is how it's moving. Initially, plus minus আছে, it comes minus plus হয়ে যাচ্ছে দেখো, it minus plus, it's changing. And at the same time behind it is repolarizing. এটা দেখতে পাচ্ছ তো নার্ভে কিভাবে যাচ্ছে? Is it visible?
Yes, sir.
Yes,
Yes,
sir.
sir.
This is how. So this is a nerve impulse moving. The electric current is moving. A change in potential is taking and it's going along the fiber. This is how the electric current is propagated. আর একটু ভালো করে ছবিটা আরেক কিছুক্ষণ দেখে নাও. Look at this picture for some time, how it's going. See, plus becomes minus, minus becomes plus, and then again when as it moves along the fiber, this again it changes back to the normal resting membrane potential. And from this nerve, this is how this electrical impulse is going. So right now in your brain, when you're reading, listening, whatever you're doing, all these nerve, I'm talking, all these nerve impulses are going. So several action potentials are going throughout my body. From my memory, it's coming. So these electrical signals are coming. So life is based on this electricity. Your movement and everything is based on this particular electricity. Is it clear? Is this
Yes,
clear?
sir.
Okay, okay.
Yes, sir.
Now let us look at an action potential. What are the components of an action potential? So in a potentiometer, you have drawn a graph. ঠিক আছে? You are measuring the voltage. Before any stimulus is given, your potentiometer is attached. It is attached one inside, one outside. Before any stimulus, you have the resting membrane potential, the straight line, resting state, minus 70 millivolts. This is the voltage and this is the time. At this point, you give a stimulus, a touch or a light or something, a stimulus is given to the nerve. Now you are seeing the measurements. The stimulus should have an intensity so much for a nerve impulse to flow. It should have a minimum amount of strength, and that is known as threshold stimulus. It should have that stimulus to send the voltage from minus 70 to minus 55. If it is less than that, there is no action potential, all or none. होले पुरोटा होबे ना तो होबे ना, अगर होगा तो पूरा होगा, नहीं तो नहीं होगा. That is known as all or none. So lots of maybe a bacteria has come and sat on your skin. It is not giving a threshold stimulus, so you cannot feel the bacteria sitting on on your skin. But a mosquito comes and pokes, immediately you will feel the sting. Immediately it will go above the threshold, and you will have this depolarization coming having. During depolarization, what happens? The voltage is changing. The voltage gated sodium channels open, and sodium concentration is very high on the outside. It comes inside. And when sodium comes inside, sodium is Na plus, positively charged. So lots of positive ions come inside. And when the positive ions come inside becomes positive. And where if the positive ions from the outside, it comes inside, so the outside becomes negative, because the positive ions from outside, they are going back, so they are attached to negative ions. So those negative ions are remaining outside, and therefore it becomes negative. And then the third phase is repolarization. So during depolarization, it goes from -70 to +40 volts. -70 volts ছিল, একেবারে উল্টো হয়ে গেছে, +40, 0 को पार करके चला गया. And again it repolarizes. Since so many positive ions have come inside, for repolarization, what happens? The potassium channels open up then, and potassium concentration is high inside, so these potassiums, it starts to go out. When the potassium starts to go out, what happens? The potassium is positively charged, so some negative charge ions are it's remaining behind, and so the inside again becomes negative like the former time, and it starts to go out so much that it becomes super negative. Hyperpolarization goes to somewhere around -90 millivolts. And then after that, the fifth stage, it comes to a resting stage. So the components are: first is stimulus, second part is depolarization, third part of an action potential is repolarization, fourth part is hyperpolarization, and the fifth part is again you go back to the resting stage. So stimulus starts the rapid change in voltage or action potential. So in patch clamp mode, so when you're putting a clamp on a nerve, my electrode inside and the grass electrode outside, on a patch clamp, it's a mechanical device. Sufficient current must be administered to the cell in order to raise the voltage above threshold voltage to start membrane potential. If it is below 55, if that strength is below 55, if it can't get it above below 55, then nothing happens. All or none. Hoga, nahi to nahi hoga. Then depolarization is caused by rapid rise in membrane potential opening of sodium channels in the cellular membrane, resulting in a large influx of sodium ions. Then repolarization, it results from rapid sodium channel inactivation as well as a large efflux of potassium channels resulting from activated potassium channels. The next is hyperpolarization. It is a lowered membrane potential caused by the efflux of potassium ions and closing of the potassium channels. সবকটা চ্যানেল বন্ধ হয়নি, পটাশিয়াম বেরিয়ে যাচ্ছে, আরো নেগেটিভ করে দিয়েছে, বন্ধ হচ্ছে আস্তে আস্তে, ইটস ক্লোজিং, বাট অল অফ them have not closed. So potassium is leaking and leaking and leaking, and it's becoming more negative. And then resting stage, when the membrane potential returns to the resting voltage. Now there are two things: absolute refractory period. There is and a relative refractory period. So during this phase, when it's in this phase, can you see this pink shaded area? Can you see this?
Yes, sir.
Yes, sir.
This pink shaded area, this phase is known as absolute refractory period. At this time, if you give a stimulus, it will not induce another action potential. মানে এখান থেকে আরো depolarization হবে না. Okay? So once an impulse is there, action potential is there, you cannot start another one. এইখানে তুমি যদি আবার কারেন্ট দাও কিচ্ছু হবে না। Okay, this current has to be given at this resting membrane potential. Only then there will be an all and none. But at this point, if you give current or stimulus, nothing will happen. And therefore, it is absolute. Even strong stimulus cannot induce. But at this point, hyperpolarization at time, this is a refractory period, relative refractory period. It is a, এখানে you have to give a হালকা না, you have to give a very strong stimulus to get some action potential. Otherwise, it will not happen. খুব জোর দিতে হবে. So these are the two phases, refractory phases. Just 1 second. I will start the sharing once more. So this is clear so far.
Sir.
Yes, sir.
Okay.
Yes, sir.
Now let us go ahead. I have not started sharing yet. Where is my nerve impulse 2? Just a moment. Part 2. হ্যাঁ এই তো part 2 আসছে। So this is the first part is over, okay? Now I'll start my sharing once more. Are you able to see this? Nerve impulse part 2.
Yes, sir.
Yes, sir.
How the electric current is moving, can you see it?
Yes, sir.
You're
Yes,
seeing
sir.
two types of nerves. This is a myelinated nerve and this is a non-myelinated nerve. Here it is coming much faster and here it is much slower. Because it's jumping, and jumping. It's not going through the myelin sheath. But here, throughout the depolarization and repolarization are taking place. And therefore, this is slow and this is fast. So all our nerves are myelinated. So this type of myelin sheath jet is known as saltatory conduction, and this type of it is known as unmodulated nerve impulse. So what are the components of an action potential? You have seen the components: stimulus, threshold, depolarization, repolarization, hyperpolarization, the resting. This hyperpolarization is also known as undershoot. I will take this next day, বুঝলে তো, on Friday. Friday evening online একটা নিয়ে নেবো, ঠিক আছে? 8 o'clock.
Okay, sir.
Okay, sir.
Okay, online.
কালকে
Friday কি হয়েছে রে ফ্রাইডেতে?
না না বলছি স্যার মানে ফ্রাইডে কালকে ফ্রাইডে তো?
হ্যাঁ কালকে ফ্রাইডে,
হ্যাঁ ঠিক আছে।
সন্ধ্যেবেলার দিকে। ঠিক আছে, ওকে
Okay, sir.
ওকে। because this is a big, आज डोज ओवरडोज हो जाएगा तब, এখানে অনেক বড় আছে, ওভারডোজ হয়ে যাবে। I don't want you to overdose and get frightened. It is not at all very tough. Kintu if you get nervous, then it will become very tough. Any questions regarding the nervous system, nerve cells? আছে কেউ?
No,
No,
sir.
sir.
No sir. Okay. Okay then, bye bye then তাহলে।
Thank you, sir.
Okay.
Thank you, sir.
You're welcome.